Hello, my name is Professor Hudson, and you are about to view a narrated PowerPoint lecture from Module 8 on biotechnology. This corresponds to Chapter 14 in your textbook. This narrated PowerPoint lecture has material compiled from several different sources, so some of the textbook photos will not be seen in this particular lecture. However, the content of this lecture does correspond to the content in Chapter 14. Biotechnology is the use and especially the alteration of organisms, cells, or biological molecules to produce food, drugs, or other goods. Biotechnology commonly uses genetic engineering to alter the genomes of bacteria, plants, animals, and humans. A tool that is commonly used and that's extensively used in genetic engineering is recombinant DNA technology. Some traditional applications of biotechnology include the use of yeast to make bread, beer, and wine, and the selective breeding of animals. Those are traditional applications of biotechnology. When we think of modern day biotechnology, we talk about genetic engineering. A relatively familiar application of genetic engineering is cloning, which is used to make identical copies of individual genes or even entire organisms. As mentioned, a key tool of genetic engineering is recombinant DNA technology, which is DNA that has been altered to contain genes or portions of genes from different organisms. Plants and animals that express DNA that has been modified or derived from other species are often called transgenic or genetically modified organisms, which is often abbreviated GMOs. So biotechnology is basically when you're trying to use living systems to create products or other goods such as food and drugs to suit man's purposes. Genetic engineering, where you alter the genomes of bacteria, plants, and animals and humans, is done a lot in modern day biotechnology. And in particular, recombinant DNA technology is a tool used in genetic engineering and biotechnology. On this slide, I'm going to list several tools used to make a recombinant DNA molecule, and these tools will be discussed in further detail on subsequent slides. So some tools used in making a recombinant DNA molecule include foreign DNA, which can sometimes be amplified through the polymerase chain reaction, plasmids, which are small circular pieces of DNA often found in bacteria, hosts, such as bacteria and viruses for propagating the plasmid in the recombinant DNA, restriction enzymes, DNA ligase, DNA polymerase, which are used for making the recombinant DNA molecule, and DNA sequencing, which is often used to check the nucleotide sequence of the recombinant DNA molecule when it's made. A recombinant DNA molecule is a molecule of DNA that contains DNA from two or more different sources. For example, if I take a bacteria gene and put it inside of a human chromosome, then that would be a recombinant DNA molecule. Or vice versa, if I take a human gene and put it in a bacteria chromosome, that will be considered a recombinant DNA molecule. On this slide, the steps in creating a recombinant DNA molecule are illustrated. In order to make a recombinant DNA molecule, the process begins by inserting a gene of interest into a DNA carrier molecule such as a plasmid. Plasmids are small pieces of DNA, circular, that are not included in the chromosome and may be present in multiple copies in the bacteria. Oftentimes, plasmids carry genes that help the bacteria survive in novel environments such as where a new energy source is available or where antibiotics are present. Restriction enzymes are used to insert a gene into a plasmid by cutting the DNA on both sides of the gene and to split it open, the circular plasmid. So one of the first things you have to do is you have to get your gene of interest that you're studying, cut it with the restriction enzyme so that the nucleotides on the ends of your gene of interest will match up with the nucleotides in the plasmid. And so you cut the plasmid and the gene of interest with the same restriction enzymes. Once you've done that, then you mix the two together, and then you use ligase to seal the breaks in the DNA so that you have one contiguous circular piece of DNA. Oftentimes when a recombinant DNA molecule is created, you have to have a source that will propagate it or to carry it and make copies of the recombinant DNA molecule. So after the gene of interest is inserted to a plasmid, it can be placed into a host organism such as a bacteria in a process called transformation. In transformation, you destabilize the membrane of the bacteria temporarily so that it will pick up the recombinant DNA from the surrounding environment. You can use that. You can either do that by using electricity in a process called electroporation, or you can use chemicals to destabilize the bacterial membrane and cell wall so that it will take up the plasmid. Once the bacteria has taken up the plasmid, every time the bacteria replicate its DNA, it will replicate your gene of interest and the plasmid that you put in. We call that cloning of the gene. When you clone a gene, you're making copies of it. Also, if there was a particular protein that you were expressing on that plasmid, the bacteria will make that protein along with the other proteins it's making without differentiating. Sometimes viruses can be used. Viruses basically are protein capsules that contain genetic material. And oftentimes viruses will attach to cells and inject the DNA into them, and so they can be used for propagating your gene of interest as well. As mentioned, polymerase chain reaction can be used to amplify small samples of DNA for use in recombinant DNA, cloning of genes, or transformation. If somebody sent you a small sample of DNA for you to study, before you started working with it, you might want to use the polymerase chain reaction to make copies of it so that you have more sample to work with. PCR is also used by forensic scientists to identify victims and criminals, and is used extensively in biotechnology and medicine. PCR involves two major steps: marking the DNA segment to be copied, and then running repetitive reactions to make multiple copies. Typically, you do the polymerase chain reaction in a machine where you add in the nucleotides, your gene of interest, and other molecules and chemicals, and then in the machine kind of simulates what the cell would do during DNA replication. And so it goes through several cycles, and each cycle of replication, the machine will allow the DNA to duplicate itself. Restriction enzymes are used extensively in genetic engineering and recombinant DNA technology. Restriction enzymes are protein molecules that recognize certain sequences of nucleotides in DNA, and they actually will catalyze a chemical reaction such that they will break the bonds of DNA at these specific locations. And this can be used when you're trying to cut open a plasmid and insert a gene of interest. The restriction enzyme can make either a straight cut, where it cuts the sequence straight down the middle, or it can make a staggered cut, generating what we call sticky ends. And this is where it cuts kind of at an angle so that you get ends with a slight nucleotide overhang. And this is another slide showing an enzyme. EcoR1 is an enzyme that always cuts double-stranded DNA at the nucleotide sequence shown. Notice that where EcoR1 cut the DNA, there's now a gap where you can actually insert a piece of foreign DNA. After you create your recombinant DNA molecule, or if you're studying a particular gene of interest, DNA sequence can be used to confirm that the DNA sequence of your gene of interest is intact and that it's in the right place in the chromosome. Also, DNA sequencing can be used to diagnose inherited disorders using sequencing in conjunction with DNA probes that can detect certain mutations within the DNA. Gel electrophoresis is another tool commonly used in recombinant DNA technology. Gel electrophoresis separates DNA segments by size. Mixtures of DNA fragments can be separated on the basis of size. Gel electrophoresis is a technique that is used to spread the DNA fragments of varying lengths in the mixture. Gel electrophoresis and the DNA fingerprints that are produced from this technique can be used in criminal profiling, maternity cases, paternity cases, mistaken identity cases, etc. This is because unrelated people almost never have identical DNA profiles. The only situation where your DNA profile would be the same as somebody else's is if you had an identical twin. We'll actually do a lab in class where we will practice running a gel electrophoresis and also reading the different types of fingerprints that can be generated from gel electrophoresis. This slide just illustrates the steps when you're going to use gel electrophoresis and labeling with DNA probes in order to identify sequences or segments of DNA. And we will do something similar to this in class when we do the lab. But basically, there are 4 steps in gel electrophoresis. First, DNA mixtures that oftentimes have been cut with restriction enzymes are placed into wells at one end of a slab of the agarose gel. An electric current is introduced to the gel. Because DNA has negatively charged phosphates, the DNA will move towards the positive electrode, with smaller fragments moving through the gel quicker than the larger ones. Thus, when you run DNA fragments through a gel electrophoresis procedure, the mixture will be separated by size. When the smallest fragments have migrated to the end of the gel, the fragments are separated into strands, and you can transfer them to a nylon sheet and stain them with a probe. Also, sometimes the DNA will be run in what we call ethidium bromide solution, such that when you look at the gel under special light, the DNA bands will actually glow orange. The techniques of PCR, gel electrophoresis, and identifying DNA sequences with specific probes have applications extending beyond forensic science. Biotechnology can be used to identify, isolate, and modify genes. You can combine genes from different organisms and move genes from one species to another. There are 3 major steps involved in making genetically modified organisms. Obtain the desired gene, clone the gene, and insert the gene into the cells of the host organism. On this slide is showing the cloning of a human insulin gene, where the human insulin gene was put into a plasmid, and then this recombinant DNA molecule was inserted into a bacteria. Then whenever the bacteria reproduce, it will make copies of this plasmid and also produce the insulin protein. This insulin protein can be purified from the bacteria and packaged and sold for people who are diabetic, who need insulin and can't make their own. Let's stop here, and in the next segment we'll talk about cloning of whole organisms, which is another technique in biotechnology.